el doctor Gatherman es de la University Cancer Center and Department of Hematology, Oncology and Clinical Immunology de Heinrich-Heine University en Düsseldorf, Germany. Él es doctor en medicina interna, hematólogo y oncólogo. Realizó los estudios en medicina en la Universidad de Heinrich-Heine en Düsseldorf, Alemania, en el Royal Free Hospital School of Medicine en Londres, en el Reino Unido, y en el Harvard Medical School en Boston. En 2002 fue profesor de medicina interna, habiendo sido previamente asistente de profesor. Actualmente es consultante del Departamento de Hematología y Oncología e Inmunología Clínica del mismo centro. Es miembro de la Professional Association of German Internists, German Society of Hematology and Oncology, and American and the ASH, and the Myelodysplastic Syndromes Foundation. Su principal actividad científica se ubica en el área de neoplasias mieloides, en particular en la epidemiología, patogenia, pronóstico y tratamiento de los síndromes mielodisplásicos y los problemas del metabolismo de hierro, desde la sobrecarga de hierro transfusional hasta los mecanismos patológicos de la sobrecarga mitocondrial de hierro en la anemia sideroblástica. Así que bueno, pues tenemos dos temas ahí importantes y por favor, doctor Gatherman.
Thank you very much for your kind introduction. Ladies and gentlemen, compared with CML, treatment results in MDS are much less impressive. However, currently, much progress is being made in deciphering the molecular genetics of myelodysplastic syndromes. And I should like to spend the first couple of slides of my talk on that subject. Now, like CML, MDS probably arises in a primitive hematological stem cell, which over many years, or actually over many decades, accumulates genomic damage from various insults. And if that stem cell, which is affected by genomic damage, is not killed by genomic damage, it may survive and actually gain an abnormal growth advantage, enabling it to establish clonal myelopoiesis. Additional mutations and epigenetic changes in MDS shape the phenotype, the clinical phenotype of the disease, which is characterized by increased apoptosis and ineffective hematopoiesis. And additional mutations and epigenetic changes also drive clonal evolution towards acute myeloid leukemia, which occurs in about 20% of cases. Now, an altered bone marrow stroma, partly as a primary change and partly triggered by the abnormal clone, together with altered immune responses, will create an abnormal cytokine milieu, which will also affect the phenotype of the disorder. Now, in recent years, quite a number of recurrent mutations have been identified in MDS. And an interesting group of mutations is this group, which seems to be related to the epigenetic changes that can be found in MDS. And these mutations seem to be related to epigenetic changes because they affect genes that encode chromatin-modifying enzymes. Therefore, most of the epigenetic changes that are observed in patients with MDS may not occur just out of the blue, but may actually be attributable to mutations in chromatin-modifying enzymes. There may be a close relationship between the genetic and the epigenetic pathway from normal to cancer, or in this case, from normal to MDS and AML. On the one hand, genetic changes can cause epigenetic changes, for instance, through alteration of epigenetic modifiers, such as histone modifying enzymes. On the other hand, epigenetic changes like global DNA hypomethylation can cause chromosomal instability, thereby aggravating the genomic instability of the abnormal clone. So we may actually get a vicious cycle. And epigenetic treatment in MDS with demethylating agents like 5-azacitidine may be successful because we are successfully interfering with this vicious cycle. Another interesting group of mutations which have been detected over the last 2 years are mutations in the splicing machinery. Nucleosome-free DNA is transcribed by RNA polymerase II to make pre-messenger RNA. And this pre-messenger RNA is then processed and spliced in the splicing complex. And this figure illustrates most of the components of the early splicing complex, as well as 3 different splicing outcomes. For instance, splicing outcome with a retained exon, a splicing outcome with a skipped exon, or a splicing outcome with a retained intron. And this figure also shows, printed in red, those components of the splicing apparatus that were found to be mutated in myeloid neoplasms and in CLL. And the idea is that these mutations somehow affect the splicing outcome in certain genes and therefore promote the development of myeloid neoplasms. Now, it has now been shown that spliceosome pathway mutations are the most frequent recurrent mutations in MDS. Overall, they are found in about 45% of MDS patients, and they affect at least 8 different splicing or spliceosome components. There are certain genotype-phenotype correlations. SRSF2 mutations are associated with CMML. And there's an even more impressive correlation of SF3B1 mutations, which are strongly correlated with the sideroblastic phenotype. But it still remains a mystery how those spliceosome mutations affect mitochondrial iron handling in a way that produces the picture of ring sideroblasts with mitochondrial iron accumulation. This riddle still has to be solved. As you can see on the lower part of this slide, spliceosome mutations are much rarer in de novo AML or in myeloid or myeloproliferative neoplasms, whereas about a quarter of patients with secondary AML after MDS have those mutations. Now, some of the genetic or some of the point mutations and cytogenetic abnormalities in MDS are virtually exclusive of each other. For instance, you can see hardly any overlap between P53 mutations and TET2 mutations. On the other hand, there are strong correlations between genomic lesions. For instance, P53 mutations are strongly correlated with a complex karyotype, probably because P53 mutations allow clonal cells that carry a complex karyotype to survive despite the complex karyotype. Now, a new dimension has recently been added to the molecular analysis of MDS by whole genome sequencing projects looking at the clonal evolution in MDS. And this is an example published by Matthew Walter and colleagues in the New England Journal of Medicine this year.